All right, in my last 10 or 15 minutes, I'm going to shift gears to clinical trials. I'm also going to catch up a little time, which is good. So this slide is not to make you dizzy. This slide puts a smile on my face. And the reason this slide puts a smile on my face is that when I got into this business, 10, 11 years ago, there were no trials. Nothing. GIPF01 was really, well, there was the Avanox trial that never got published in terms of industry-sponsored trials, and there was nothing. And this just shows you the activity that we've been having. Now, most of it's been negative, but some of it's been positive, and we'll talk a bit about capacity 1 and capacity 2 here in a little bit. But the variety of work has grown dramatically. And I'll touch a little bit on the endothelin receptor antagonists, which are currently active, and the IPF net studies. Now, you all know that the gamma studies have since conclusively been proven to be negative. And anybody on gamma should not be on gamma. It's a very high-cost drug with no proven utility at this point. But there are a lot of other interesting studies, including the anticoagulation trial in the Kubo study, that showed a positive result in survivorship and laid the groundwork for one of the IPF net trials as well. But this is terrific. We're still way behind the cardiologists at 32,000 patients in a trial. But at least we're looking at in numbers that let us actually see something. And that is a huge step forward. So what's active, rather than going through what's been done? Well, the Build 3 trial finished enrollment some time ago and is awaiting the end of its event-driven design. Starting October 1, I believe, the PRED-AZA-NAC or Panther study will be open, looking at change in FVC, and I'm going to go through that in more detail. The sildenafil trial, looking at sildenafil, and I'm going to show you some data in both directions on that. We enrolled 182 patients, looked at 6-minute walk distance, and the database is locked and will be reviewed shortly. The anticoagulation trial I've kind of touched on. The endothelin receptor antagonists, triple angiokinases are being looked at. Senecor is looking at an anti-CCL2, which is currently actively enrolling. Early phase developments, phase 1s, GC1008 took a look at a TGF-beta modifier that is technically much stronger than pirfenidone and gives me room for excitement in regards to the pirfenidone results about what might come down the road. Treprostinil, which was the Cotherix trial, was stopped for safety reasons. So let's move into the capacity trials, which are probably what everybody's most interested in. And this data was presented at ATS and is currently under review by the FDA. There were 2 concurrent, nearly identical, multinational, randomized, placebo-controlled trials. 110 sites in 11 countries. Patients were randomized to oral pirfenidone or matched placebo for 72 weeks. Over 770 patients. That's just awesome. We looked at different treatment regimens, and the eligibility requirements were a confident diagnosis of IPF in the not too severe range. In other words, your DLCO was above where the threshold is for survivorship in that first year, and the FVC was meant to reflect that. The primary endpoint was an absolute change in percent predicted FVC. Now, I'll tell you, they designed this before Ron Dubois published his data, but what it basically says is, if we can decrease the rate of decline in the FVC, which I've shown you is a poor predictor, but the rate of decline is a good predictor, that can be used as a surrogate for mortality. You see, they spent $70 million on GIPF07 as a mortality study. That's a lot of money for a small biotech to spend. So the question is, can you have a more efficient design that will give you the same information as a surrogate measure to try to figure out the same thing? And it's as reasonable a design as you can think of, reasonable enough that the IPF net used exactly the same design for Panther in terms of an endpoint measure. What did they see? And this is fascinating because the Japanese studies were positive. In looking at the percent predicted of FVC at 72 weeks, capacity 1 was clearly negative with a p-value of 0.5. whereas capacity 2 was clearly positive at a p-value of 0.001. What do you do with that? And the answer to that is, we're not sure. If you take a look at the data in a little more hardcore fashion, what you see is that the rate of decline in the pirfenidone group was the same in both studies. But the rate of decline in the placebo group in capacity 2 was higher than placebo group in 1. And if you look at the 48-week mark, you can see that it would have been a positive study. Now, this is an issue of spin. I can tell you my personal opinion about this, which is I think the drug works, just not well. And the reason is I think that that's a reflection of power. I think if you do it longer and more, you'll see a difference. And what's an acceptable difference is for all of us to decide vis-a-vis the risk-benefit of the drug. But it is a question of spin, because one could make the argument that, gee, it worked at 48 weeks, and if you kept taking it, eventually it just starts to wear out its usefulness. I mean, that's one way to spin it. So it can be spun one way or the other. It's not a definitive answer. And what the FDA will say is anybody's guess as well. But I wanted to show you what the hardcore data actually looks like so that you can understand, if there are reservations in anybody's mind about what it does or doesn't do, why it's there. Now, there's more interesting aspects to the data. If you take a look at progression-free survivorship, which it's not powered to look at, right? It isn't long enough to look at that. It was a 1-year study, 72 weeks, a little over a year. And it's really a reflection of that same FVC decline. It's a direct reflection. The p-value was 0.023 in capacity 2 and 0.355. Again, it's a question of the number of events. In capacity 1, you have more events in the placebo group because more of those patients declined by 5 or 10% on their FVC over that same time period. And that's how that surrogate marker works, right? And that's where the reflection is of that data. But you can't look at this as a primary endpoint. Now, what gets interesting is they then tried to break it out by categories. I was actually going to skip this, but the bottom line is it reemphasizes the point I made. It doesn't matter how you slice it. If you start to break the patients into groups by 10% decline or 20% decline or improvement, you end up with the same p-values. Capacity 2 is positive and capacity 1 is negative. Tells you that that decline in FVC over time is a decent predictor. What is interesting, and Paul Noble presented this material, is that there's a change in the 6-minute walk distance in capacity 1 that we don't see in capacity 2. So the drug's probably still doing something. It just didn't get reflected in the FVC measure at that measure in time. My interpretation of that, as I said, is a power both of the drug and a power of the size of the study, which is why I flippantly make comment that it probably works, just not well. And then looking at the overall survivorship, again, no difference in the hazard ratios.